The brain has always fascinated me already since high school. And when I started my neuroscience studies, I realized that animal testing is really valuable, but that I also don't want to work with animals. What I also realized is that I wanted to do something of societal relevance that can improve health of humans and the environment. And neurotoxicology was the topic where these two interests came together. In order to test the safety of chemicals and pharmaceuticals, laws require information that you can often only get from animal tests. And for us humans, that's useful because that means that we generally have safe products. But what scientists increasingly realize now is that animal tests are not always predictive for humans, especially when it comes to the brain. And also, tests for brain development are not mandatory in the current testing requirements. So from an ethical perspective, you can also say, is it okay that we sacrifice so many animals for something that's not necessarily predictive for humans? That's why a lot of scientists, including me, are working on animal-free innovations. For example, on cell-based tests that we can use to do toxicity testing. And in our lab, we work with stem cell-based models, one based on mouse stem cells and one on human stem cells. A major challenge with these in vitro tests is that they can only mimic a small part of human biology. So you can see it as a little puzzle piece, and we need a lot of these puzzle pieces together to form an organ. And in the case of brain development, which is super complex, you can imagine that we have to integrate a lot of these in vitro tests together that can mimic these biological processes in the human brain. So for my research, I looked at the biological domain. And that basically means that we looked into which biological processes are present in our in vitro tests and which are not. And we did this by looking at the cells under the microscope, and we also looked at gene expression. We also tested chemicals, and these chemicals, we know that they trigger certain biological processes. So if you see that in your in vitro tests, for example through a change in gene expression, you're pretty sure that that part of brain development is mimicked in your in vitro test. And that gives you a bit of a clue of how your puzzle piece looks like. So what about our in vitro tests? How do these puzzle pieces look like? Well, so for the mouse stem cells, we know we have stem cells that we differentiate into what we call neuroprogenitor cells. And from these neuroprogenitor cells, we differentiate them into nerve cells and two types of support cells. The human stem cells are already a bit further in development. So they start as a human progenitor cell, and they are differentiated into nerve cells and one type of support cell. And importantly, these nerve cells make connections with each other, which is a really critical event in brain development. To even better define what our puzzle pieces look like, we tested compounds on both in vitro tests. And a good example of this is fluoxetine. So on the mouse stem cells, we saw that fluoxetine triggers gene expression changes in the nerve cells, as well as in one of the support cells, the oligodendrocytes. We couldn't see gene expression changes on oligodendrocytes in the human stem cells because the oligodendrocytes are not there, so that's logical. But we did see the same gene expression changes in the nerve cells in the human stem cells, and we saw that specifically the connections were affected by fluoxetine. And this is consistent with existing human and animal data. You can imagine that when we test more and more compounds, you get a better picture of your in vitro test. What can it do? What can it not do? And thereby you can see what the shape is of your puzzle piece. So that's something we need to keep working on. Another thing we need to think about is which other puzzle pieces we need. So which other cell-based tests we need to connect with these tests. And then if we have all of that, we also need to think about how to integrate everything. For example, using computer models. And then hopefully in the end we have something like an animal-free brain with which we can test the harmfulness of compounds on brain development. We need everyone, from scientists in the lab to industry to regulatory bodies and actually the society at large to really embrace animal-free innovations and set them as the new normal.